Welcome to Biology Made Easy. In today's lesson, we are discussing how to solve genetic problems. And this is for O-levels and secondary school grade 10 to 12. Welcome. We start with a very simple question here. Mating pure strain black-bodied fruit fly, Drosophila, and pure strain gray-bodied strain, the offsprings were all gray-bodied. The offsprings were allowed to interbreed and they produce offsprings, some of which are gray-bodied and some black-bodied in proportions of 3 gray and 1 black. When you read genetic questions, there are clues. So in this one, mating pure strain means we are dealing with a homozygote and we decide to use a letter A, then it means we are dealing with some individual with either big A, or small a. That's pure strain. So mating black-bodied, so we have black-bodied and gray-bodied, and the offspring were all gray, meaning gray is dominant over black. So the parents will be big A, and small a. Otherwise, you won't have all gray. So all gray, you have big A, small a. This is the information we get from this line. So these offspring were allowed to interbreed, means this gray and gray, and these were F1s. So these were F1s, they were allowed to interbreed. And they produce offspring, some of which are gray-bodied. So the information we have here is we have offsprings that are gray-bodied and offsprings that were black-bodied. This offspring is F2. We have gray-black, and in proportions of 3 gray, so we have 3 here, and 1 black, 1 here. This is the information that we get from this. That is how to analyze genetic questions. Put the information in genetic terms. So now let's get to the questions. How many genes are involved? In this, we are talking about 1 gene. 1 gene, because we talked about black-bodied and gray-bodied. Only 1 color, body color. So the name of the gene is body color. It's 1 gene, body color. How many alleles are involved? We looked at 2 alleles. You have black body and you have gray body. So the alleles are 2. And the name of the alleles are black body and then gray body. Those are the names of the alleles. And then we are supposed to explain the results of the experiment with genetic diagrams. This is what we are going to do, explaining the results right from the beginning here, where black-bodied strain mated with gray-bodied strain. Parent phenotype, we have black body mated with gray body. And then parent genotype, black body is homozygote recessive, and gray body is homozygous dominant. There will be meiosis, gametes will be produced, random fertilization takes place, and we have all of it will be gray-bodied. That's the F1. And these F1 phenotypes, all gray-bodied. So here we have all gray here, and we are supposed to solve the F1s. So selfing F1s, we have parent phenotype, we have gray-bodied, parents genotypes, all gray body, meiosis, gametes, and we want to use the Punnett square to make things easy. So you have to learn how to use the Punnett square instead of crossing like this. So the Punnett square, we have male gametes up here. This is the male gamete. And we have our female gamete down here. The female gamete also like this. So we have this individual, this individual, this individual, that individual. This is gray, gray, gray. So we have 3 gray, 1 white. We have 1 white, we have 3 grays. Let's attempt the second question quickly. This question is very long and you should not be scared with long questions. Just read and take note of clues and write them down with a pencil. Seeds develop inside pea pods after fertilization. They contain starch. A gene, so we are dealing with a gene. A gene controls the production of an enzyme involved in the synthesis of starch grains. This results in seeds that are round. So we are going to deal with round seeds. We can have any letter. The allele, small r. So round is round here. The allele is small r here. The allele small r does not code for enzyme. The starch grains are not formed normally. This results in seeds that are wrinkled. So this allele produces seeds that are wrinkled. The diagram shows round and wrinkled seeds. So you have round seeds, wrinkled seeds. All this information, looking at it can scare you, but you can break it down. Now, pure breed plants, pure breed, we are talking about homozygotes for the gene concerned. Pure breed can be big R, or small r, small r. This is pure breed, this is pure breed. So you write the information down. A plant breeder has some purebred pea plants that are grown from the round seeds. So we have round seeds, big R, and purebred seeds that are grown from the wrinkled. So this is what the plant breeder did. And there's a question here. State the genotype of the purebred plants that are grown from the round and from the wrinkled. Very easy. This is the genotype of the round. And then we have genotype for wrinkle here. Because we started analyzing right from the beginning. Then these purebred plants were cross-pollinated. What this one is saying is that, and that was said to be cross 1. So purebred plants, you have round here, and this round were crossbred, and they said that is cross 1. Still analyzing, and the seeds were collected. All the seeds were round. So from this information, we see that what we got out of here is round. That's the F1 kind of. So they are round, but they are heterozygous round. Then these round seeds were germinated. So the seeds that were collected from it germinated, grown into adult plants, and said to be offspring 1. So this is offspring 1. And self-pollinated. So let's go back. These round seeds were germinated, grown into adult plants, right? That's offspring 1. And self-pollinated, cross 2. So this cross 2 is this. So this is selfed. This F1 plus F1 is selfed. And that becomes cross 2, self-pollinated. Let's continue reading. The pods on the offspring 1 plants contain both round and wrinkled seeds. So what came out of here? We have round seeds and then also wrinkled seeds. We have round and wrinkled seeds. So this is it. Further crosses 3 and 4 were carried out as shown in the table. So that is the table you need to fill in. We have cross, we have phenotype of seeds in the seed pods, we have round seeds, wrinkled seeds, we have ratio of round to wrinkled seeds. And we need to fill this table. We have to use a tick mark if seeds are present, a cross mark if seeds are not present. All right? And then also we need to state the ratios of round wrinkle. Now, the first box has been filled already. You have purebred round seeds crossing with purebred wrinkled seeds. That is this purebred round with purebred wrinkle. That's cross 1. So you see cross 1 here. The result we had is all round. So you have round seeds, no wrinkled seeds. The ratio 1:0. So how was this ratio gotten? We have round, round, crossing with wrinkle, wrinkle. That's this one. That's here. And we have F1 all round. What really happened is round, round, gametes is round, wrinkle, gametes. Using the Punnett square makes things easy. We have gametes round, round here. This is the male. The female gametes will be here. So we have this offspring will be this offspring this offspring like this offspring like this. We have all individuals are round. 1, 2, 3, 4 round. So 4 round, 0 wrinkle. And the individuals are supposed to be 4 from each cross. So we have 4/4 round, 0/4 wrinkled. The ratio is 1. Here, the ratio is 0. That's how come this ratio was obtained. Now let's look at cross 2. We've been told there are round seeds, there are also wrinkled seeds, and we will have to produce the ratio. And the cross is offspring 1. This is offspring 1. It's round like this. We are self-pollinating it. So round offspring 1, round offspring 1. Gametes, using the Punnett square, we have our male gametes here, female gametes on this line. And so we end up having offspring. This gamete and this gamete will produce this offspring. This one and this one produce offspring. This bigger and this one will produce, and this one will produce this offspring. So we have these individuals. This is round, this is round, this is round. So we have 3 rounds. This is wrinkled, 1 wrinkled. And there were 4 individuals. So the rounds will have 3/4 rounds, and we will have 1/4 wrinkled. So the ratio of round is to wrinkle. We have 3 wrinkles and 1 round. Now we have cross 3. We have offspring round cross with pure breed round. So we have this offspring 1. This is the pure breed round, and gametes will be this. Using the Punnett square to cross the gametes, we have the male gametes here. We are choosing this as a male. We have big R, small r here. We put our female gametes here. Fertilization takes place, this and this will unite. We have big R, big R. The phenotypes, all this is round, 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 round. So we have round, we have 4 rounds, and 0 wrinkled. So the ratio will be 4/4 for round, 0/4 for wrinkled. So we have a ratio of 1:0. So we go to fill in our table here. Here we are. We have 4 rounds. So there will be round seeds. there will be no wrinkled seeds. The ratio is 1:0. Now we have our question 4, and offspring 1 with pure breed wrinkled seeds. Here is offspring 1, that's offspring, and with pure breed wrinkled, small r, small r. So gametes, we have this gamete and that gamete. Punnett square, our males are here. If this is the male, we have big R, small r. The female is on this side. We have, this is the female, that's the female, so small r, small r. Fertilization will take place. We have big R, small r, small r, small r. So we have phenotype 2 round, 2 wrinkled, and we have 4 individuals from a cross. So the ratio of this will be 2/4 rounds. The ratio of this will be 2/4 wrinkles. So we have half here, half here. So in actual fact, the ratio is 1:1. So let's come to fill our table here. The results, we have round seeds are present, wrinkled seeds are present. The ratio is 1:1. The last question, which states that seed shape in P is an example of discontinuous variation, suggests one reason. The reason is simple. P shape is determined by major genes. We will only have 2 genes. You have round, stroke wrinkled. And this is discontinuous variation. If the P seeds were determined by polygenes, that many genes were involved, then it would have been continuous variation. But it's not. It's determined by major genes. Well, this is where we end for today. If the lessons have been useful, please give us your like and do subscribe. In our next lesson, we'll look at symbols that will be used to solve genetic questions before we do other questions. Thank you very much. Goodbye.